there was Larry, you asked, you mentioned the CRD10 gene. I wanted to know whether there is any developmental component to that reaction to bananas. I mean, like, did he always hate bananas, or when he was a child, he might have basically, like most babies, like gobbled them up, you know?
I did not fully hear your question, but he's had it since he was a child. He hoped it was over, but it's not over yet.
Do you think that when he was a baby, he ate bananas?
Oh, I'm not sure about that. I should ask him. Yeah, that's a good question.
In general, babies eat bananas.
Yeah.
I have never heard about a baby that doesn't eat bananas.
Yeah. Yeah, our baby loves bananas.
Our two babies too. Only anecdotal evidence, but still.
Yeah.
I'm using a microphone, that's why I'm being loud here.
No, of course, but I think it should work. Okay, so keeping on this path of sensory reception, so the manner in which they turn the sensory world into the neural code is very similar to ours. So this is actually one of the first times where whole cell recording was used in the C. elegans. This is out of Corey Bargman's lab back in 2018, actually only a few years ago. And they showed that there is actually spiking in these neurons, similar to the action potentials that we fire. And they showed that if you increase the gradient on this same sensory neuron that we've been talking about, if you increase the gradient of stimulus, you see a little burst of activity at every gradient step increase. So this neuron is detecting a change in the amount of this chemical, and then that will be translated down into the ultimate response. So if the response is really strong, then that will tell you you're basically right on top of the signal. If the response is weak, that will tell you you're cold but getting warmer, and you need to keep looking for the source of the signal. And so this is using electrophysiology, and they find that there's quite a lot of calcium-mediated action potentials, not as much sodium-mediated action potentials as we have in the mammalian system. And if you record with GCamp, which I don't know if everyone here has talked about or been told about GCamp yet, but that is a genetically encoded calcium signal, fluorescent signal. And basically you can make it so that the neuron will literally light up when calcium increases. And that is a nice indirect signal of activity of the neuron. So GCamp imaging is a fairly common neuroscience tool now, where you can genetically express GCamp in neurons and then image them in real time, video image them, and watch them light up as they receive some sensation or perform some behavior. So certainly I will show you those in later lectures, because GCamp is pretty cool. So it's definitely worth looking at. Okay, so where are we? So the reason I include the electrophysiology in the testing causality moment is that this process of removing sensory receptors, or of eliminating neurons maybe, or deactivating some part of the body and watching the response while the animal tries to do the same behavior is not the only way that you can test causality. You can also record from the neurons during the behavior. And in this case, you record from this AWA sensory neuron, you present diacetyl, and you see a response. And so therefore you know that there is a causal link between that sensation and that response. Presumably that response is then used in the normal processing, because the brain doesn't just respond for nothing. Every signal that's going on within the brain will be incorporated into the state of the circuit and incorporated into the decision making and ultimate response. Okay, so in order to test causality, though, there's obviously also genetic manipulation. If you remove certain receptors, or if you kill certain neurons, for instance, if you kill the AWB neuron or the AWA neuron, and then you give the odorant on one side of the field and control some sort of like saline or something else on the other side of the field, you can track and see how the animals respond to these things. So if you kill the AWB neuron, what you see is that the animals tend to stay in the middle here. If you give no odorant, or if you give odorant, and if you kill a different neuron entirely, then you don't see that same response. You see that this neuron is not involved in detection of that same odorant. But so killing these neurons is very easy in C. elegans because we know exactly where they are, and you can literally just zap them, or you can genetically remove them, knowing which genes are actually involved in the incorporation of it. So I would recommend you check out this paper too. It's a very clear example of testing causality with genetic manipulation. And here's a better image of it. So in this case, you have the AWA neuron with this odor 10. You have an odor 10 mutant with no receptor, or you can now artificially put odor 10 into the wrong neuron. So the AWA neuron is typically an attractive neuron, and the AWB neuron is typically a repulsive neuron. But if you artificially move the odor 10 receptor onto the AWB neuron, it will now respond in the opposite way to normal. Okay, so this is another way of testing causality. This YouTube link right here will show you a video of this. If I remember correctly, this is a lecture from Corey Barkman, and it shows very nicely how you might do this. And so this is the sort of thing that you'll have to think about if you're designing a network. You'll have to remember that every one of these neurons would be has to be used for a specific purpose, and it has to be wired up to the correct neurons to ultimately produce the right behavior. Okay, so I'm going to skip ahead. I'm just going to show you this one to show you that also they have negative phototaxis. So right now a light is going to turn on and the animal moves away. So these are animals that live deep in the dirt. If they detect light, that means that they're near the surface, and that means that they can be eaten pretty easily. So they actually view light as an aversive stimulus. So if you show these animals light, they will move away from it. It was not known for a long time how these animals actually can see because they don't have eyes. They don't have what we would think of as typical visual receptors. And then someone figured out that they not only can see, but they have specific UV frequencies that they respond to most that are associated with the most powerful UV signals from the sun. So it makes sense that they would be tuned to those signals. And no one knew quite for some time how they actually were doing that. What was the real receptor that allowed them to detect light? And what someone found in 2016 was that it was a mutated taste receptor that had mutated to allow for the detection of photons of UV light, which is really quite strange. Now, if you look up, for instance, how we see ourselves, we see in 3 primary colors. Blue, green, red, but you might know that there's a frequent color blindness of green, red, especially in males. And you should really investigate why that is. It's very interesting. And it's quite similar to this mutation, where something starts as the receptor for one thing, and then over time, a mutation will allow that receptor to now detect something else. And there can be issues with the kind of holdover of that kind of the classical response of that receptor. So in this case, a taste receptor is now a UV light receptor, but there could still be some sort of interference of the taste receptor on this receptor as well. But this is how the C. elegans is actually able to see because they don't have eyes, as you can imagine. But a mutated taste receptor allows them to actually see. So no need to remember this part, but this is the complicated step for phototransduction. And the C. elegans also has a very similar to us. So this is actually the phototransduction series in mammals. And what you can see is that you imagine that the detection of light should be quite rapid, but light actually evokes this very complex intracellular cascade of molecular changes that ultimately activate a light receptor neuron. And this is very similar to what is going on in that mutated taste receptor in the C. elegans. But so I've just included this to note the process of detecting light, and we'll talk more about that in just a little bit. Why is this going there? Okay, so I've included just a few videos. I'm not going to go through all of these. I'm going to show you this one so you can understand what the rest of them are. This is using neural activation, using optogenetic stimulation. Optogenetic stimulation is a relatively new tool where light activated receptors are genetically expressed in neurons such that you can turn on a light and activate the neuron and then watch what happens basically. In this case, the animal has these channelrhodopsin receptors throughout its body. And what you'll see is the turning on the light of the field around the animal, and it doesn't do anything. And now the light just on the animal, and it paralyzes the animal. So in this case, you have a situation where if you activate all the muscles at once, the animal can't move anymore, because obviously locomotion requires a complex series of well-timed activations and inhibitions of different muscles. For instance, you can't move your arm like this unless the muscle back here is relaxed. And then to move your arm back down again, this muscle has to be activated and this muscle has to relax. So that's what's constantly going on when the animal is moving. So in this case, if you express channelrhodopsin throughout the animal and activate them with light, you will paralyze the animal because they're not able to move in sync anymore. So the rest of the videos that I've included in here are very similar, but I'll post this presentation and you can see those different examples. So you see on this one, for instance, the animal can continue moving the rest of its body, but not the little block where you're turning the light on here in the midsection of the animal. So this is just another way of testing for an actual causal relationship between the neural network and the ultimate behavior. Hopefully you can remember all of these, and you might even be able to think of more of them. But you have genetic manipulation, where you alter the expression of channels or you alter the literal existence of specific neurons. You can record from the neurons during a sensory response or the performance of behavior, or you can change the activity of the neurons and see how that changes the behavior. So these are some examples of how you would actually test for causality and know that what I'm telling you is accurate. So those videos are in a series there, and when I post this lecture later on, you can go back and see more of them if you want. I always find them quite amusing at the very least, and they're pretty interesting. Okay, so that light receptor that I was explaining before, the same thing was done in order to actually test for causality to see if that was really the receptor for how the C. elegans can see. They were presented with a UV light, and their locomotion speed was tracked. Oh, come on. Sorry, everything's getting in the way here. When they actually manipulated that that light receptor, they could track the locomotion of the animal. So in this case, what they did was they put that light receptor into a neuron that was responsible for moving the animal, and they turned on a UVA light, and they suddenly were able to stop the animal from moving because of the expression of that presumed light receptor. And again, remember that the point of this paper was that they did not know how C. elegans could see. They didn't know of a good receptor in the C. elegans for detecting light. So this was their way of testing whether or not this was the right receptor for it, because everyone had called this previously a taste receptor because it clearly was a taste receptor at some ancient point in the C. elegans existence. But over time, it mutated into now what could be a light receptor. and it actually was specifically primarily a UVA light receptor. So that's another example of testing causality. And this is the sort of experiment that is backing all of the circuits that I'm showing you in order to prove that those are based in reality and not just sketches that anyone could make. I think Julian has a question.
Maybe I missed it.
Yeah.
But I don't actually know what locomotion means.
Oh, sorry. Yeah. No, that's a good question. So locomotion is any sort of movement in a directional way. So moving your whole body in space. So we use locomotion typically in humans is walking, but a locomotive, also in English, could be a train. It's any sort of movement. So locomotion is a general term that allows me to speak about movement of a creature like a C. elegans and a creature like a human and a creature like a fish. So all of them are using drastically different sets of muscles and neural circuitry and just kind of physical movement in space. But the goal is to get somewhere and move your body in space in a targeted and organized manner. So in this case, the C. elegans locomotion is them wriggling on the agros or in the dirt that they live in. In humans, it's walking towards the cookie. For a fish, it might be looking for the bug that's on the surface. They would locomote in that direction. So locomotion is any that general term at all. So just kind of movement of your body in space.
Thanks.
Kansu, you have a question also?
Yeah, so you said that like they knew this receptor as like a taste receptor before, and then they realized that it was actually also like a light receptor because of a mutation. So did they also, when they realized that, did they also keep testing it for if it's still a taste receptor too? Like, can both be at the same time? I don't really remember if that can be the case.
In this paper, they suggest that it is no longer functioning as a taste receptor, but I would always be hesitant, I guess, to say for sure that it wouldn't function as a taste receptor anymore. But the reason that it would have been considered as a taste receptor would be because you can map the genes that produce these receptors, and genes have a clear kind of family organization based on the structure of the gene itself. And so you would clearly map onto the taste gene family. So that's why it wouldn't have been known as a light receptor in the beginning, but they were able to detect that this receptor was detecting light, I think because they eliminated it. I think they were testing it as a taste receptor. They eliminated it and they found that the animals were no longer responsive to light in the same way. And that led them down this path of realizing that there was a light receptor. I think that was the origin of this paper. But now I'm blanking on, I have to look it up. I think they did a little press release about it and gave a kind of a background of it. But I would assume that there might still be some taste function, but they were suggestive in this paper that there was no longer a taste function, that it had mutated sufficiently to only be a light receptor. But you can always double check these things.
Yeah, thank you.
Okay, so. All right, so now we're going to kind of move on to no longer living in this very simple world of the C. elegans, but now we're going to take on the complex world of the mammal. And I say simple world of the C. elegans, obviously that's not entirely true. The C. elegans is itself very complicated. So then if you imagine a human or maybe a mouse, you can already see how much more complicated that's going to be. But that is what we're interested in understanding, is how the brain controls behavior, even in this complicated scenario. So I've put here kind of a definition of sensorimotor integration, because that's the general thrust of what we're talking about today, is the process of producing the right motor output based on sensory information, and often sensory information from multiple modalities. And a modality would be different types of sensation. So Vision would be one modality, smell would be another modality. And so if you see the word modality, that's what it's talking about. If you see multimodal integration, they're talking about both seeing and hearing something and making a response to the combination of those two senses, those two modalities. So you might see that terminology throughout this topic as well. Okay, so now we can start talking about how you and I perceive the visual world. Maybe you've already seen a structure like this, I think, but just to remind you that you have the eye, you have the series of how your retina is organized. You have the lateral geniculate nucleus and the thalamus, that is the kind of the first stop for vision, and then you have the primary visual cortex. There are other components to this, of course, that we'll get into, but this is what you need to know for now. And basically, that is the first processing of light, then the actual literal turning of light into the neural language. Then you have these low-level feature processes and then high-level feature processes. And what I hope you can appreciate is, if you imagine the cookie scenario again, where you see cookies, you remember that that's a good thing, you stop what you're doing and you move toward the cookie. What you have to realize is that at this point, you may not even be at the level of deciding that it's a good thing. You're only at the level of beginning to detect what you're actually looking at. Once you get to the visual cortex, you have not even begun to make the decision of how to actually move towards the cookies. So the number of steps in the brain, especially of a brain like the mammalian brain, is really high in order to allow for the ultimate proper detection of the visual field in front of you, and then make the correct motor response in response to what you're looking at. Okay. So I don't think they've gone over this stuff.
No, but they have talked about on and off cells.
Okay, cool. Okay, so then some of that will be familiar to you, but this is the kind of the first stages of what you might look like. This is a kind of imagined reality of what your at the level of your retina, what the neurons are seeing, what they're actually detecting, and that's because your brain is designed to detect kind of the easiest things around you in the world. And the easiest thing visually to detect is edges, is actually lines of contrasting image. So on the left here, you obviously see a little girl with a bunny, and it's actually quite a complex series of different gradients of light. And of course, this is in black and white, and in the real world, you would see it in color. But then here, what you're going to immediately detect is you're going to fade out all of those differences in the gradient of the signal, of how intense the light is, and you're just going to notice big changes in contrast. So her shirt or jacket versus the black background, that's a really rapid or a really sudden change. And so you're going to first of all detect this edge between it. So you're going to detect the edge here, and you might start to detect the more subtle edges, like the bits of her hair, and obviously the difference in the eye of her toy bunny here. But this is your immediate processing. So if you want to turn this back into what it actually is in reality, we're several steps away. So the first thing you're going to see is a detection of the contrast, the edges. And part of how this optical illusion works is because of your brain's obsession with edges, and it's using it in order to detect motion. So if you imagine what motion is, you imagine that it is a movement of light across your retina in a series of steps. So as I move my hand and you're looking at my hand, that is sending a signal to your eyes that is actually sending specific light across your retina and detecting the change from here to here to here to here in a sequence. And that is converted in your brain as motion. Now, something like this optical illusion in front of you can trick your brain into thinking it's moving because of these series of high contrast lines that are shown in front of you, that is activating your retina in a sequential way, or it feels like a sequential way. So if you're looking directly at one of these circles, so if you look right at one of the dots, you'll see that there's no illusion of movement, but all of the space around where you're looking, there is indeed an illusion of movement, because you're making these very subtle saccades that you can barely even detect, and that's moving. Saccades are just movements of the eye. We'll get into that later on, but you're making these tiny little movements of your eye, and that's actually driving this detection of these kind of light and dark lines that are on the image in front of you, and it's making you think that you're seeing movement. And one of my favorite ones is this one, which I think is a very intensive optical illusion. And this one really, to me, is very mobile. You can really see that there's quite a lot of activity on this image in front of you. Yeah.
And there's a Mandelbrot set in the middle.
Yeah. So this is a little nod to the fractal images.
Fractal plans.
Yeah. So so again, this is the way that this illusion works is very straightforward, and I think illusions are useful for understanding how the mind actually works. So if you understand that this is an error, basically, the fact that you're detecting movement is incorrect. Your brain is incorrectly detecting something in the world. But it's important to remember how it gets there, because it's fairly straightforward. And it's important to also then remember that your brain is always making this essentially a guess of reality. It's got a series of algorithms that it's running based on these specific inputs. And most of the time that's correct, because most of the time you're not going to bump into this image in the world. I mean, the fact that we can make this image is basically an indication of human advancement. I mean, it's very complicated that we can even make something like this. You would never bump into such an optical illusion out in the real world before we had digital art or any ability to make something like this. So in general, your brain works very well at detecting reality. And the way it detects reality, we've figured out ways of tricking by using an optical illusion like this. And in fact, there's also audio illusions that it had never even occurred to me until recently, but Aaron Wong showed me that fairly recently, and I'll try to remember to show you that at some point. Very interesting to imagine that you can trick your auditory system as well in a similar way to this sort of optical illusion, but I'll show you that in the future. So this is detecting motion, and the way you're detecting motion is, again, by looking for contrast and sequential series of activations across your retina. So detecting objects is a similar thing. So as you saw in that first image with the little girl holding the bunny, you were able to find the edges. You then noticed a clear pattern of a face and a bunny. It's actually quite a complex image that you saw there, but you were able to pick out all the components of it because you have experience with the world and your mind went through all of the steps in order to ultimately get to the recognition of the visual field in front of you. And so what I hope you can see here is that there's actually several horses standing on a snowy mountainous backdrop. And in order to detect that those horses are standing there, you have to first detect the orientation of lines. So you might have noticed these legs, which are quite high contrasting compared to the rest of the snowy landscape here. You can detect the color. You can detect, obviously, contrast. You can detect disparity. This is a two-dimensional image, so it's a little more complicated, but if you were really looking at a three-dimensional world, you would notice that one horse is standing farther back than another horse. To some extent, we can experience that now, even though it's a two-dimensional image, we're kind of experiencing a faux version of that because of the perspective trick that we can play when we make a two-dimensional picture like this. And then obviously movement direction is another way that you can detect objects. Nothing's moving here, but if they were walking, that would make it extremely easy to detect that there's a horse there and it's not just a landscape. So that's really low-level processing. And when I say low-level, that's actually already a couple steps in. But that's the lower level processing. And then once you get to next level processing, you can now start to, for instance, follow along. If you find an edge here, you can follow along and identify that that's part of one piece of a leg, that's part of a whole, that's part of a whole object. You can then follow it up into the animal and realize, oh, not only is this a single leg, but this is actually a single horse. The whole thing together creates one object. And that means that you need to follow these contours. You need to detect the text, the kind of the imagined like textile shape of the surface that you're looking at. You can look at what is part of the horse versus what is not part of the horse, even though it's quite similar. So all of this has to take place at lower level processing. And then it's actually many synapses down the road until you can really say, okay, that's a single horse. That is a single thing. I know it. I even might remember riding one at some points. I might remember seeing one on holiday, or if I saw one as I was walking around the countryside, I could see a horse. So you can not only identify this object, but now start to have all these memories and associations with it. But all of that takes quite some time to get to. So the neural network for looking at this image and then getting to this, even though it might have been very quick for you, required quite a lot of steps. So early parts of that is what we're going to talk about today. We're going to talk about moving our eyes around. So keeping in mind the horse image that I just showed you, why do you think I'm now moving to control of the eyes, of actual muscle movement of the eyes? Does anyone have an idea of how that would, or why that would be relevant for detection of a horse on that image?
Because you need to move your eyes around the picture to see.
To see what?
To see the contours of the horse.
Yeah, so you have to now kind of follow, and you will first kind of scan the image, and then you'll begin to notice contours, orientations, different colors, different contrasts. And the only way that you could do something like contour integration is to actually move your eye along and put your fovea, which is at the center of your eye, which is the highest density of really high resolution image detection. You have to foveate, if you will, on different parts of the object or different parts of the image in order to follow along the object. Exactly. So that means that not only are you going through this whole visual process in order to ultimately get to the horse, but there is an active motor component to seeing in order to even look at an image like this. So you can imagine it's obvious that there's a motor component as you look around in the world, but there's also a motor component to looking at something directly in front of you, making these small movements of your eyes in order to pick up on all these elements and actually give your brain these this series of visual information in order to ultimately put it together into an object. So we need to control our eyes exactly so we can look around the image. So this is a classic example of a picture of a woman in a scarf or some sort of head wrap, and you have taken actually more than likely this series of movements around this picture in order to actually look at what's in front of you. And you may not have even cognitively or consciously noticed that you were doing that, but when I first presented you this picture, in order for you to determine the object as a whole, you have to look at individual things, and you are surprisingly not able to detect in high resolution things just outside your fovea, just outside of your direct line of sight. So you really have to move your eyes along, and we can do this really, really quickly. And we do it at this point almost automatically because we're developed humans, but this is not something that comes innately. You have to develop the motor skills in order to do this. And you have to recognize really quickly some element of this picture in order to start following the trace of looking for a face. So what you'll first do almost always is look at the eyes if you're looking at a face. So you look at the eyes, and then you'll go to the mouth, maybe back to the eyes again, and then the outline of the face. So that's what you're seeing here. So this is the way that we would detect a face. We have actually neurons in our brain that are facial recognition neurons, that once you get to the point where you've identified this as a face, those neurons are activated. But you have to first give your brain all of the visual information necessary by moving your eyes around this face in order for those facial recognition neurons to actually light up. Okay, so as I said, that's not something that always takes place. So early on in development, this is what you're seeing with these red dots is where the baby is looking. So the baby is primarily looking at motion of the mouth in this scenario. So initially they might look at the eyes, but then as the woman is talking, a baby is going to be really obsessed with the thing that's moving. And later on, we will also be obsessed with something that's moving, and we will be looking there, and you are probably looking at her mouth too. But of course, that's layered on top of the fact that I'm showing you this video with these dots, and so you're probably following them around. But also at this point, you will be able to take in the full scan of her face, eyes, mouth, ears, and full outline of the face, whereas a baby will pretty early on learn to do that, but initially be obsessed with the actual moving item on the screen. So that's something that you can see in development. This is an example of an 18-month-old versus a 30-month-old baby. And what you can see between the two is that they are actually looking at similar but different things. The fixation number is higher with development and the fixation duration is lower. So with time, we learn to scan the visual field very quickly and take in a lot of information very rapidly, whereas really young children or babies cannot scan things so quickly. There's a lot of motor coordination that has to be developed. There's a lot of recognition of items that need to be learned. And so they will take more time and they will move more slowly between different objects in order to actually see what they're looking at. Okay, one more example from children. So this is a fixation point that the baby is looking at. And so what you can see here is the ball was rolling. It went behind this box and the baby's eyes stopped. And then they pick up again when the ball comes out the other side. So this isn't a 5-month-old infant. They've not yet really learned about object permanence. Once a child gets to a certain developmental stage, they will already have jumped ahead, expecting the ball to move out from behind that box. So this is just a matter of the coordination of understanding the sensory world and moving your eyes in a proper way to detect the sensory world that has to be learned in development. Okay, so this is a classic video. I want you to count how many times the players wearing white jerseys pass the basketball, and then we'll discuss it after the end of the video. Hopefully the video will be will play properly. Okay, so keep in mind the white jerseys, count how many times they pass the ball. What's that? Yeah. So you're having to track with your eyes and make a lot of movements and really stay focused. Hopefully you got the correct number of passes, and you might have seen the gorilla or you might not have seen the gorilla. I don't know if you noticed it. So walking into the middle of the video was just a gorilla. And so this is a reminder of,
choppy, so they might have seen it.
yeah, so if the video is too choppy, it might have messed up my experiment here. But that's a classic example of attention and the integration or the kind of the combination of sensory motor integration that's needed to correctly attend to something, but it's also a reminder of how much you need to really be directly looking at something to properly notice it and to properly perceive it. And we have this kind of sense, because our brain is very good at tricking us, that we're perceiving the whole world all around us all the time, but we really are only perceiving what we're directly foveated on. And then your brain is making guesses, statistical guesses about everything else around you as you scan the space. You obviously have peripheral vision to some extent, but it's much less good than we might imagine. So you have to keep that in mind. And that's why you really need strong control of your eyes in order to really see properly. So multiple types of eye movement that we're going to run through. Are information gathering eye movements like saccades or smooth pursuit or vergence, and then stabilizing eye movements like the optokinetic reflex and the vestibular ocular reflex. So these are all things that you do all the time. You may not even be aware of them. And I would be surprised if you were aware of the neural circuitry of them, but we're going to talk about them today. We're going to talk about how different the neural circuitry is in these things that are essentially just the same sort of movements of moving your eyes around. But remember that your goal is different in these different eye movements, and the specific sensations that you're receiving in order to drive them is very different too. And so that means that you have, to some extent, different sensory receptor neurons that are responsible for ultimately feeding back to the movement of your eye, and you have different intentions to them. Some are more conscious and some are less conscious. Okay, so this is how complex the muscles that control your eye are. Obviously, you certainly can, maybe you've never thought about it, but your eye is a sphere, so you can recognize that your eye is a sphere, which means that it has quite a lot of degrees of freedom, not, a full freedom. It's not like your eye can spin around in your skull. There's a limit to how far it can move. It can really only move in this number of degrees, but quite a lot of different directions. So you can move all around. You can roll your eyes, look up, down, right, left, or any combination in between. And you can move your eyes together or farther apart, depending on if you're focusing on something close to you or something far away. So if you imagine that you look at your computer and then you look up at the wall behind your computer, you had to take your eyes from being closer to being farther apart so that you could keep your fovea directed on the same thing, but now farther away. And that's vergence, and we'll talk about that a little bit later. But all of these movements are controlled by this complex series of muscles. You have these two muscles on the side that pull your eye either left or right. You have two muscles on the top, the superior rectus and the inferior rectus. So the rectus is just the muscle that will directly pull the eye. And then you have the oblique muscles that allow for more rotational movements of the eye. So the eye is controlled by one of your cranial nerves. You have 12 cranial nerves. One day, maybe you'll have to memorize all of them. It's been a long time, but there was at some point a mnemonic that I had to use to memorize these for neuroanatomy tests. I no longer have to think about the cranial nerves all the time, but at some point that was a very important part of my life. But we will talk today about the ocular nerves, the optic nerve, which is the sensory nerve that goes directly into cranial nerve 2. That is receiving all of the visual input of your eyes. You've got one on this side and one on this side. And then you've got quite a complex series of cranial nerves that are involved in all of those, in control of all of those muscles that are controlling all of the movement of your eye. So you've got the trochlear nerve, number 4. You've got the ocular motor nerve. You've got the abducens motor nerve. Facial nerve can sometimes be a component of this, but it's not a direct eye movement nerve. But so you have both sensory and motor cranial nerves, and they are either receiving information or sending motor control. So these are the motor control cranial nerves, and this is the sensory nerve with respect to the eye. And then obviously you have all of the rest of them that are receiving other information. Another one that we're going to get into today is the vestibular nerve that's running here to the vestibular cochlear sensory nerve. And the reason we'll get into that is that there's a strong interaction between your vestibular system and your visual system. And I'll define what vestibular is in just a little bit, because that one is less well understood than vision, I believe. Vision, I think, is a little bit simple to understand. It's simple to conceive of. Okay, so these are the actual nerves that go to your brain, or I guess flow out of your brain in order to receive sensation and control the eye. And this is how that process actually looks. So once you start to imagine how your eye is turning light stimulus that reaches the retina into the ultimate vision that you see on the screen in front of you, you have to realize that even the very first steps of that are complicated. You have to realize that you're looking at a world that's spread out in front of you, and you're receiving that information from a little sphere that is your eye. So what you have is the signal on the right side of your eye here and on the left side of your eye. So if this is your left eye, then you're receiving information from the, let's call it the nasal side of your eye and the temporal side of your eye, because this is your temporal lobe over here, and obviously your nose is here. And those are very different bits of information. So just because it's a signal coming from this eye doesn't mean it's all from the left side, because the side on your nasal eye is actually detecting information from far on the left, and the side on your temporal side of your right eye is detecting information from far on the left, because you have to remember that this is a sphere that is detecting light in these opposite directions, right? So if you're looking at a pencil, the way that works is obviously light comes down and bounces off the pencil. And then the top part of the pencil is going to come down and cross your, oh yeah, sorry, I'm losing track of myself here. And it's going to come down and be on the bottom of your retina. And then the bottom of the pencil will actually come up and be on the top of the retina. So that will be the same for left and right. Information on the left side will actually come into your, cross your pupil and your iris and be on the right side of your eye. And so that means that you have to keep track of these different directions. And the way your brain does that is it actually makes those separate tracks. And so the nasal side of your right eye will cross to the other side and meet up with the temporal side of your left eye, and vice versa. And then ultimately you will have organized in your visual cortex, which is here in the back of your brain, you'll have it organized properly in space. Okay, so we're going to start walking through the processing vision and how your brain detects these things. But first, we're going to take another break. I'm probably going to get another coffee. And then, but if anyone else has any questions, then feel free to ask. Otherwise, we can come back at 11. And then we can wrap up with some of the sensory motor integration stuff that we need to talk about. Cool.
Very nice.
Let me know if there's any questions.
Did you address Kate's comments
Oh.
at 10:38? I
I
don't know.
might not have. Let me see. Oh, is this pattern different for people who have trouble looking at eyes? Oh, I see. That's complicated. Because that's, we don't know if that's a sensory issue or. Let me think.
Yeah, I think it's complicated. It's a very good answer.
Yeah.
You know, whenever there are too many influences, then you get
Yeah.
noise in the system.
Yeah.
The noise expresses itself in various ways.
Yeah. I'll think about that while I get the.
That's a good one.
Is there a machine just here?
There is. I have something to show you.
Okay.
Just press the button. Should I, if it has lights?